Well, thank you very much, and thank you for inviting me. I've been invited to this meeting every year, and last year I wasn't able to be here, and I figured out, but I was really pleased to hear that there's a lot of new families here, a lot of new faces. I see a lot of faces I know, but I'm glad to hear that there's a lot of new families here, because what I'm going to do is give a little bit of history about the deletion syndrome, as well as go over chromosomes and, you know, what does 1p36 mean. And that's the one thing I wanted to start with is if you want to sound much smarter than your doctor, it's 1p36, and I'll go over why that is, and not 1p36. I'm sure a lot of your doctors have called it 1p36. But before I get started, so after I speak, Bernice Morrow is going to speak. Bernice and I have collaborated on a lot of different projects, and one that we've been collaborating on for the last few years is 1p36. And so Bernice and I have been working with the cell lines that I've been creating. So when you make cell lines, you get fresh blood from the individual, and then you put it with a virus that makes it immortal so that you can continue to grow it forever. And Bernice and I have been using those cell lines, and we've collected some of these cell lines in 1994, I think, or 1996, I think, were some of the first bloods that we got in. And as you'll see in her talk, she's doing expression analysis, which is really, really important. And when we go to publish this data, we may get criticized because we're using immortalized cell lines, which can, you know, you expose them to this virus and they can change. And so what we want to do is replicate some of the experiments with fresh blood to prove that what Bernice has seen in her lab is real and not a reflection of the cells that we created in the lab. So what we're going to do, and you'll hear more about her research, I'm going to pass around a pad, so if you want more information about her research, this is a way that Bernice can then contact you after she gets back to New York. And then all of protocol. Some of you that were here 2 years ago, I talked about how important it is that if you're going to participate in research studies, that, you know, it's approved by an institutional review board and that you are properly consented and all that good stuff. Well, this is a IRB approved research project. So if you want more information about it, please put your name on this and we'll pass it around, and then Bernice will contact you with more information. Okay, so let's get started. No, I don't know how to use this. Oh. So why do I get invited to speak a lot to various groups, including your group? And the main reason is that chromosome abnormalities are really common in our population. They are probably common in other animal species as well. But for the most part, those animal species probably, they don't survive, and so we don't see them. But we see them in humans because we have medical care. So about 0.5% of all newborns actually have a chromosome abnormality. So it's very, very common in our population. And actually, chromosome abnormalities are the most common cause of developmental disabilities and birth defects in our population. So sometimes you hear about single gene disorders. You might have heard the term point mutations. It actually is mostly our problems with our chromosomes that cause medical problems for us. And deletion 1p36 occurs in about 1 in 5,000 newborns. So this is one of the most common deletion syndromes. It's actually probably the second most common deletion syndrome, and it's the most common chromosome syndrome that happens at the ends of the chromosomes. And I'm going to show you chromosomes, and we'll talk about the different parts of the chromosome. So you'll hear the word, and you've heard it before, the word syndrome. And what does syndrome mean? Well, syndrome basically means it's just a group of signs and symptoms that occur together and characterize a particular abnormality that we see in our population. And traditionally, new syndromes have been identified by a very astute physician who sees a patient with certain clinical features, and then maybe a year later they're like, you know, I think I've seen this before. And then they spend time going through their files. You know, this was all before we had electronic records. And then they start collecting patients and comparing the patients. They say, yes, these patients all have a group of symptoms or medical problems that are the same, and now I'm going to call it a syndrome. And many times the genetic etiology or the basis, why does this syndrome occur, was not known. Usually the clinical syndrome was described way before the reason for that syndrome was identified. And the most classic syndrome is Down syndrome. It's a little boy with Down syndrome. He was actually part of my PhD thesis. And Down syndrome was first, as a collection of medical problems, was first described by Sir Langdon Down in 1866. And it's quite remarkable that it was almost 100 years later that the etiology or the reason for Down syndrome was identified, and that's the extra chromosome 21. Now, this is, do I have a pointer? Yes. Do I know how to use it? There. So this is Robert, and these are not Robert's chromosomes. Does anybody know why these are not Robert's chromosomes? Huh? Because he's a boy. And this is a female. There's 2 X chromosomes. Yay, you guys are all cytogeneticists now. You didn't realize that before you had kids, but now you're all cytogeneticists, right? So then in the 1980s and the 1990s, more syndromes started to be described. And sometimes now, because we had better methods in the laboratory, the syndromes were now described based on what we were seeing in the lab. And now one of the classic examples is 1p36. So, and I'll go through how we discovered 1p36, but here, I like this slide, and you're going to see it again. This is a collection of individuals, some that were in my study and some that just contributed their photographs to me so that I could show. And I'd like to show this picture, and some of you have seen it before, because even among different ethnicities, you can see that there's some very common features among these individuals. And so we'll go through some of these features, and you guys all know what they are. You know, the very straight eyebrows, the wide-spaced eyes, the little pointed chin, small mouth, and some other features. So when we first started describing chromosomal syndromes, what was needed is that they had to be big. So in general, many of these were described that were 10 million base pairs in size. And if they were smaller, we needed other methods in order to see them. Now here's a pair of chromosomes 1, and this one is deleted. So we're looking at the 1p36 is the little white fluff at the end. Very, very hard to see through the microscope. I put an arrow on the one that's deleted. Otherwise, it would be very difficult to see the difference between the 2 chromosomes. So the first report in the medical literature of a deletion of 1p36 was in 1980 by Hain et al., where they had described a child with an unbalanced translocation that involved some other chromosome in the genome and then also involved the very end of chromosome 1. Now, after that, there were several other reports, but almost all of these reports included children who had unbalanced translocations. And the reason for that, and I'll show you some examples, is when you have a big piece of another chromosome stuck on another chromosome, which is what a translocation is, it's more obvious. You can see it. And so that's why these first cases had what we call unbalanced translocations. And I'll go through. It's hard to talk about one thing without showing the other thing, so my slides are a little unorganized in that respect, in that I'm going to go through the different kinds of chromosome abnormalities we can find. So translocations are one. And so that's how these first reports came about. So again, the translocation allowed us to be able to see it through the microscope much easier. So, and as I already told you, this very light staining band, or that little white fluff at the end, makes it very hard to see through the microscope. And if we look at all the literature from the first, cases for about 10 years, there were only 14 patients of 1p36 reported in the literature. So there really wasn't very much. And then we started seeing deletions. So let's go through the chromosome, and then we'll talk about what happened when I was at Baylor College of Medicine. So this is chromosome 1, and I just wanted to orient everyone to the structure of the chromosome so that we're all talking about the same thing. So this is the short arm. There's debate in the literature why the short arm is called the P arm. Some people say that the French named it for petite, but then when you ask, well, then why is the long arm named the Q arm, they say, well, because it's the next letter in the alphabet. Well, that doesn't make any sense. And it really, if you talk to a Frenchman, it doesn't make any sense to them either. Others have said that it's because it's a mathematical equation that P plus Q equals 1. Remember all that from algebra? And so that's how it got named. We don't really know for sure, but the long arm is called the Q arm. And the middle is called the centromere. And the centromere is a very important part of the chromosome because it's what makes the chromosome move during cell division, because all of our cells have chromosomes, or almost all of our cells have chromosomes in them, and it's important that the chromosomes get from the parent cell to the daughter cell. And it's through the centromere. Now, at the beginning, I told you that 1p36 is the most common terminal deletion syndrome. It's not the most common deletion syndrome, but it's the most common terminal deletion. What I mean is, it's the most common deletion syndrome that involves an end of a chromosome, because the end of the chromosome is called the telomere. So you might see in some of the things that you're reading that's a telomeric deletion. And again, the white fluff at the end is 1p36. The very, very last band is 1p36.3. And the one is subdivided into even smaller bands. So based on 1p36.1, 0.2, 0.3, and then 0.3 subdivided into 0.31, 0.32, and 0.33. The fact of the matter is, when you're looking through a microscope, it's almost impossible to differentiate these various bands. And I know that most of you who have had chromosome reports, the cytogeneticist did assign a particular band, but if you gave your child's chromosomes to 5 different cytogeneticists, you're probably going to get 5 different interpretations of this region. Hopefully they'll all see it, but the banding is not very accurate. And so what's the most accurate test you can have is a microarray. And if your child hasn't had a microarray yet, there may be a way to get that done at no charge. And I think it'd be important for you to have that done. So you can, let me know if your child has not had a microarray study, and we can see if we can get that done. Because it's much more accurate. It's going to tell you how many millions of base pairs of DNA are missing. And again, this is fairly arbitrary how these numbers are assigned when a cytogeneticist sees the chromosomes. Okay, so when I was at Baylor College of Medicine, I was the director of the cytogenetics laboratory. And starting in about 1993, we started seeing this deletion on the short arm chromosome 1. And we went to the literature, and there wasn't much in the literature. There were a lot of unbalanced translocations, but there wasn't really a lot of information about this deletion. And within 3 years, we found 14 cases, which was the same number that was already published in the literature. And it's like, well, why is our lab, I don't think there's anything special about the air in Houston. Why is our lab finding so many microdeletions? And I started going to scientific meetings and presenting these cases, and other cytogeneticists thought I was making it up. Because they're like, I've never seen one of these. Cytogeneticists is much older than me. I've never seen one of these in the lab. And, I just had to tell them, I frankly believe that you're missing them. And it turns out that they were missing them, because this is very, very common. So these first 14 cases were all identified using routine chromosome analysis, where we look at those banded black and white chromosomes, and we looked through a microscope. Now, during this time period, we looked at the population birth rate in Houston, Texas, and in one year during this time period, there were 60,000 births in Houston, and we found 6 deletions of 1p36. So we thought that this frequency could be, as common as 1 in 10,000. But then we realized that half of these kids had more than one chromosome analysis. So the 1p36 was missed on the first chromosome analysis, or karyotype. And because it was missed, we figured that, if we found 6, there's probably 6 still out there that got missed, because we're all looking at chromosomes, and there were 3 or 4 other cytolabs in Houston, and they hadn't found any. So then we thought, well, then it could be actually as common in 1 in 5,000. And this is the number that we now quote, and we believe that it is this common. So these are the kids. These are the first 14 kids in the study. And some of these kids actually may be in the room because they're going to be about how old, Callie? 24. That's right. So, you know, if you see yourself up here, let me know. She was 4 years old at the time.
No, she's patient number 4.
Oh, she's patient number 4, yeah. So, Callie, isn't she cute? But how old were you in... She's about 4 or 6, yeah.
6 when I was diagnosed.
Okay, 6. Okay, yeah. So these are kids for your study because they're all adults now. Yes. So then what we started doing was a clinical review, and that first paper we published was really a clinical review of these 14 patients so that we can understand the features that were associated with the syndrome. And the facial features, as I told you, are very consistent among kids, and I'm sure that you all have noticed that here. And by publishing this kind of medical literature allows other doctors then to hopefully recognize the syndrome. And we're hoping that is what is happening. And I actually have met families who said that the clinician suspected it before sending the FISH or the array test. So it is getting out there. So the next thing we did was 2 years later, we collected 30 patients for our study at Baylor. And then we started to look at the molecular characterization of these deletions. And one of the things we found, which we've never been able to explain, is we could look at the deletion to see whether it came, they're all new. All of these are new deletions. They happened either in egg or sperm. Okay, so the parents don't carry the deletions, but we could tell where it came from. We could tell whether it came from the egg, something happened when the egg was being made, or we could tell whether it came from the sperm. And what we found is that we had more deletions that came from the mom. And this has continued to hold up as we look at this, and we don't quite understand why. We know that egg and sperm production are very, very different. You know, I don't know if you guys know that when baby girls are born, they have all the eggs they'll ever have when they're born, and those eggs sit there until the girls go through puberty. Whereas boys don't start making sperm until they go through puberty. So some people think it's because the egg is sitting there for 13, 16 years or however long. But we don't really have any evidence for why we're seeing this. So it's still very interesting to me. So we could determine the sizes of the deletions because we now had new tools. We didn't have to look through the microscope anymore. We could look at the DNA. And again, as I said, we had more maternally derived or deletions derived from the moms than we did the dads. A frustration for me, and this is now 20 years, has been that we don't have a good correlation between deletion sizes with certain clinical features. We know that when you have really large deletions, and it doesn't matter if it's in 1p36 or somewhere else in the genome, really big stuff tends to cause more problems. But what we were finding is that kids with 2 megabases or 2 million base pairs missing or 4 million base pairs missing or 6 megabases, they tended to have the same features, but they weren't necessarily consistent. Some of the kids had seizures and some of the kids didn't. Some of the kids walked and some of the kids didn't. Some gained speech, some didn't. And it's been a huge frustration for us in trying to figure this out. And Bernice will talk a little bit more about it because we now have better molecular tools and there may be ways to figure out why we're not getting a good correlation. Because I've published for many different syndromes, not just 1p36, and in some syndromes we can find the gene that's causing the problem, and in 1p36 it's been very difficult. So what we did was we then had enough cases to publish what we called a physical map, where we actually aligned all the chromosomes, and I'm going to show you some schematics of that, again, to try to characterize the syndrome. So by 2003, we had 61 patients enrolled in our studies. And by 2008, we had over 130 patients enrolled in our studies. Let's see, what's the next one? And again, it's the most common syndrome. So I have probably either enrolled into my study or communicated with over 300 families with 1p36. And that's probably just the tip of the iceberg. So I suspect that one day, instead of 50 families being here, which as I heard has registered, which is fantastic, you guys are going to have to find a bigger boat. Because it's, as the information grows, as more people find your website, there's a lot more families out there that probably don't know about these meetings, and so the meeting could get quite large. So one of the things we really realized once we started getting into all these different samples is that the kinds of rearrangements that cause deletion of this region varied between families. And so some of you have seen this schematic before, but it just shows all the possibilities. So the first is a terminal deletion where you have a break at the end of the chromosome, and then the chromosome somehow heals because the ends of the chromosomes are protected by this little tag, the TTA-GGG, and this tag protects the chromosome from further breakage. So somehow these chromosomes heal. Again, it's not well understood, but some of your kids have what we call terminal deletions, where there's a break, one break, and everything distal to that is gone. And then there's another kind of deletion that's called interstitial, where you have two breaks. And the end of the chromosome now is capping that chromosome. And so some of your kids have interstitial deletions. And then some of the kids have what we call a derivative chromosome, where there's actually an end of a different chromosome on here that's capped that. And that's usually due to a translocation. And that's why many of you, when your child was diagnosed, the physician then wanted samples on the parents, the two of you, because they wanted to make sure that one of you didn't carry a kind of rearrangement that can cause 1p36, which is called a translocation. And then finally, we had a number of cases in our laboratory that had complex rearrangements, and some of your kids might have these, where we had multiple breaks in the chromosome. So something happened. The chromosome sort of blew up, either in the egg or the sperm, and then when it got reassembled, there are missing pieces and sometimes extra pieces. So we actually have in the study some individuals with very complex rearrangements on the end of the chromosome. So again, this is how a terminal deletion happens. We have one break in one chromosome, and then everything distal to that break is missing, and then the chromosome somehow has healed. And then an interstitial deletion, again, is two breaks, and then this end of the chromosome comes back on, and now the chromosome's shorter. It's missing some material. And then a translocation, shown here, where you have a break in one chromosome and a break in another chromosome, and these two pieces. So carry one, that's why, again, that the physician wanted to check your chromosomes, make sure you didn't have something like this, and I'll show you why in a moment. So in this particular instance, with a balanced translocation, you have all your genetic material, it's just that it's rearranged. And the problem is the cell doesn't know that. So when the cell goes to make eggs or sperm, it needs to make sure the right partners are going together. So in this example, this chromosome without the translocation and this chromosome without the translocation have gone together. And so this offspring would be what we call normal, but what's normal? But anyway, they have the right chromosomes. And then if if you look at the blue arrows, if both translocated segments go together, then the offspring or the baby, again, is balanced like the carrier parent. So they have all the material just rearranged. But again, the cell doesn't know how these chromosomes are supposed to separate. So then sometimes what can happen is the wrong pairs go together. You get the normal chromosome with one of the translocated chromosomes, and now you're missing the hatched piece, right? You guys following this? And then you have extra of the solid piece. Does that make sense? And then the opposite can happen. You can get this chromosome where now you have extra of the hatched piece and missing of the solid piece, which is what we see in 1p36. If you imagine that this is the chromosome 1, you've got some extra material here and you're missing 1p36. So are there any questions about that? And I'd be happy to talk to you individually afterwards if you want to tell me, if you have questions about your individual chromosomes. So terminal deletions, interstitial deletions, and unbalanced translocations can be identified on the microarray. Whether or not one of you carries a balanced rearrangement where all the DNA is there, it's just been rearranged, can't be determined by a microarray. And in that case, they either do usually fish, or chromosomes. Yes. Good question. So let's talk a little bit about microarray analysis, because I know it changed my life, and in some ways I think it changed some of your lives, because it really, when we started using microarrays for diagnostics, it was the first time in my life where I got letters and cards from parents, and then once we started all using email, or emails telling me how grateful they were that we were now using microarrays, because they had been searching for a diagnosis in their child. I think the oldest I received a card from was a 21-year-old who, it wasn't 1p36, but they had been searching 21 years for a diagnosis, and it turned out to be chromosomal, and we can identify it with the microarray. So what is a microarray? Well, there's different kinds. I'm just going to show you one kind here. And with this particular microarray, what we do is we take control DNA, which is usually a DNA of someone that we know is chromosomally normal, so either 46XX female or 46XY male, and we extract their DNA. And then we take the patient's DNA, and we don't know what their chromosomes look like. That's the question we're asking. And we want to compare these two DNAs, and we want to see, is the patient, do they have extra or missing DNA compared to the control that we chose to use? And so we label the control in one color, which is red, and we label the patient in a different color, which is shown here in green, and then we put them together on this microarray. And basically a microarray is just a solid support. In the beginning, we actually just used a piece of glass, and we've spotted the human genome on it. And when I first did microarrays at Baylor, we spotted just the end of chromosome 1 on it, so just 1p36. So it's very small. And what we're asking is each one of these spots, which represents a different region of either 1p36 or a different region of our human genome, is there extra or missing material in the patient? That's basically what we're asking. So, if it's balanced or if there's no gain or loss of material, then we're not going to see it. So we're just looking for gains or losses. And luckily, we don't have to really look at these spots. If the patient actually has extra material, the spot looks more green. And if the patient's missing material, it looks more like the control, so it looks more red. But we don't actually look at it. We use computer analysis, which is great. So what we have here is the whole chromosome. And you think about it as it's laying down on its side with the short arm over here and the long Q arm over here. Here's the centromere. We don't have probes to the centromere because the centromeres among chromosomes are very similar, so we don't put the centromere on here. And then we look for a difference, and here we can see that this individual has this blue dip here, and all these probes go down, which indicates a deletion. And so now we don't have to look at those black and white chromosomes. We can simply see very quickly that this patient has a deletion, and it's terminal because it takes out the whole end of the chromosome. And then we can do FISH, where we take a green probe to the centromere and a red probe to 1p36, and we can see that this chromosome has both the control and the end of 1p36, so it's normal. And here's the deleted chromosome. It's got the green control, but the red is missing. And this is an example of an unbalanced translocation, where again we can see that 1p36 is deleted. This is a much bigger deletion compared to the last one. And now we have a gain on chromosome 5. And this is very typical for an unbalanced translocation where 1p36 is missing, and this piece from chromosome 5 is now attached on 1, which you can see here. So the arrow shows that there's chromosome 5 material on the end of this chromosome 1. So this one shows the deleted chromosome 1, and this shows that there's a normal one because we're actually taking material from 5, and so it's on 5, and it's on this 5, and it's also on the 1. So that's an unbalanced translocation. So now we have new technologies that we can use to identify cases of 1p36. And because the microarrays scan the whole genome, and a lot of times the specimens come into the laboratory with just developmental delay or seizure disorder, it's very, very rare that a case comes in that says suspected 1p deletion. It does happen, but it's pretty rare. Usually it comes in labeled with some of the symptoms that the child's having. So it is really an unbiased estimate of the frequencies of the different chromosome abnormalities we have in our population. So we started testing at Signature Genomics in 2004 using microarrays. And during that time, we identified 179 new cases. So these are new cases. These are not part of my research study. These are cases that were sent to the laboratory because the child had developmental delays or seizure disorder. These, and some of you I know have had testing at Signature, and that's how you're identified. So your child is counted in this 179. Oh, and Ken?
What percentage of microarrays do you think your lab does?
So what did we used to do? So what happened was, when we started in 2004, no one else was using microarrays diagnostically, so we had 100% of the market. But the market was pretty small because people were kind of worried about using it. You know, they weren't sure about this thing. Which was kind of a stupid business decision, but we decided to teach others how to do microarray analysis. It really was. I mean, it wasn't a stupid business decision because not everyone's going to send their cases to our lab, right? So I wanted to teach others in the country, including Cincinnati Children's. We taught them how to do microarray analysis. And then these labs started doing it themselves and created a lot of competition in the U.S., which that's fine. I'm over it. Because now we collaborate with all these labs because they're also identifying 1p deletions, right? At the height of among the competition, we were doing about 35% of the microarrays in the country. Now we do much less. We have much less market share because there's so many labs doing it now. So, but we've done, I mean, I've lost track, probably 60,000 microarrays in our lab. It's a lot. So if you look at the 179 new cases identified, we see that about 50% of them are terminal deletions, one break, everything's lost. We see about 35% interstitial deletions. About 6% of the cases have these complex rearrangements where we have multiple gains and losses, and about 9% unbalanced translocations. And it's really the 9% that we all need to keep in mind. So when a new family contacts you and says, we just got a diagnosis, you should say, well, I apologize for prying, but did you guys get your chromosomes looked at? Because it's really important, because if a parent carries a balanced translocation, they can, it can cause the unbalanced form, and so they can have multiple kids, with 1p36 deletion, or they can have the reverse, and they can have kids with the unbalanced translocation that involves the other chromosome too. What we've been doing is looking at the terminal deletions compared to the interstitial deletions, and I'll show you some of that data, to try to narrow the regions in which to search for genes. And so that's actually helpful in that they have different breakpoints. What we tend not to do, we published on the complex rearrangements because we were very interested in kind of the molecular mechanism, like what happened to this chromosome that it's got gains and losses, and sometimes it's the pieces that reinserted or inverted, and they're really crazy chromosomes. So we published on those because we kind of wanted to understand, what happened to this chromosome, but they're not very helpful in mapping the clinical features. And then we stay away from the unbalanced translocations. Again, we published on the unbalanced translocations because we're interested in why did this chromosome exchange with this one. Why did the translocation form? But it's more of an intellectual curiosity. We don't use those to look at the clinical features because these kids not only have deletions, but they have gain of material from somewhere else in the genome. And sometimes they'll have, it's hard to tell which clinical features are caused by the deletion versus having extra materials from those kids. So I was just going to briefly go through the clinical features, but I think Dr. Hopkin is really the one you should talk to. But I'm just going to show you the kind of the frequencies of things that have been published. If we look at the typical craniofacial features, we see that about 77% of the kids have the typical features. And these include, kind of a small head, the epicanthal folds, which are the eyelids of the eyes. They can have sort of low set ears. And what was interesting to us is that they can have very large late closing anterior fontanelle. That's a soft spot. And probably some of you remember that, your kids had very large ones and they're closing. And we published a paper, it's been a while ago, where we actually had kids with duplications and triplications of this region, and they actually were born with the sutures fused. So it was very interesting to us that you actually have this opposite medical problem, whereas you have deletion, you have wide open sutures, and if you have duplications or triplications, it actually, the children are born with that already fused, which is not a good thing either, because the reason why it's open is for the brain to grow, so you don't want to be born with it already closed. Now, as far as neurologic, this kind of gives you an idea of when we talk about, neurology, these are the kinds of things we're looking at. Oh, I forgot to give you my disclaimer. By the way, I'm a PhD, not an MD, so any medical questions, you can ask Dr. Hopkin. I forgot to give that at the beginning. So intellectual disabilities or developmental delay, pretty much, 100% of these kids have that, but you know what's really difficult is if they don't have that, they probably don't come to medical attention. So is it really 100%? Are there kids walking around out there who have this deletion and they're doing great and they haven't come to medical attention? We think that, and we have some evidence, that you can have really tiny deletions in this region and it's probably not causing problems. And so those, if we can identify those people, they're good to know too because it excludes regions for us to look at, right? If you can have a deletion and you're doing just fine, then we don't look in that region for genes, for problems. But so far, because these kids come to medical attention, we say 100% have some sort of intellectual or developmental disability. Hypotonia, as you know, is a problem. Speech delay, nonverbal speech in about 64% of kids. And, again, these numbers are based on what we see and what Dr. Battaglia has seen in the kids that he's examined. And it differs a little bit from study to study, but it kind of gives you an idea that, not all the kids have seizures. And not all the kids have, feeding difficulties. So it just kind of gives you an idea of things to look for. Now, as far as heart anomalies, there may be other heart anomalies that we just hadn't seen, so it didn't get in our group, but those are shown here. The non-compaction cardiomyopathy seems to be relatively common among the heart defects, but again, 71% of the kids have some sort of structural heart defect, so it's really important for these kids to be seen by the cardiologist. We used to think that this was much higher. Some of our early publications had a much higher number, but as we look at more and more kids and our numbers get bigger, we see that it's about 50% of the kids have some sort of hearing loss, and again, about 50% of the kids have vision problems. But that's certainly a high enough percentage that you certainly want your kids checked out. And gastrointestinal, if your kids have any of these, I'm sure you guys are well aware of it. It seems to be, a common complaint of these kids. Now, we do see MRI abnormalities, but that's really common among chromosomal syndromes in general. There can be changes that are consistent with the epilepsy. This is an abnormal gyration. You know how our brain has all of those little gyrations in it? This is really, really small ones, and it's considered not in the normal population. And then there's many other things that can be seen by an MRI.
Sort out.
And when do you have to stop taking them to that particular specialist? Yeah, no, it's a great question, and that's what this study at Cincinnati is going to answer, because we certainly lost kids to follow up. So we weren't the initial, 60-some kids that enrolled in my study when I was at Baylor. We were not able to follow all those kids. So some of those were lost to follow-up. My biggest problem is I had doctors that were lost to follow-up. So I couldn't get a hold of the original doctors, that were part of the original studies to continue working on it. So I'm really happy that this is going to happen at Cincinnati Children's, because we need centers that want to dig in and really understand this condition. And it's my understanding that Baylor is becoming interested again in doing this work. But that's been the hardest part to follow up. It's not just losing the families to follow up. It's keeping the doctors involved in the studies. Because it's really important to have consistency in the studies. So we used to have, some of you saw Dr. Shapira, and he saw every single one of those first 60 kids, and that was really helpful. So why do we want to identify the genes that cause the features in the syndrome? First of all, it gives us a better understanding of the cause of the clinical features, because we're curious individuals and we want to know what's causing this or that. It's also important, if we can identify and we know the size of your child's deletion, then we can anticipate certain medical problems. And right now, that's really hard to do, because we have kids with very small deletions who have seizures, and we have kids with very large deletions that have seizures, and that's not very helpful. And for me, the biggest reason for wanting to do this research is to identify targets, meaning genes, for possible treatment. So some smart drug company could actually make a drug that interacts directly with that gene and can treat some of these symptoms. And so that's my hope. And we're a long ways off from that. And I can't believe I'm saying this 20 years later, because I really thought this research would be a lot easier. We're a long ways off from that, but I think we're going to get there, and we'll identify at least some of the genes that cause some of the features. And then hopefully there's a way to treat that and enhance the child's life. So I just wanted to show you, how do we map these regions? Well, this example is the cardiomyopathy. What we do is we align all of the kids' breakpoints. The blue line are the terminal deletions, and the red lines are the interstitial deletions. And then we look to see what's the smallest region of overlap between all of these deletions. So we take, here, this deletion ends here, and this child has cardiomyopathy, so we can say, okay, well, this is the largest it could be, and then what's the smallest, and that's actually the breakpoint here. And what was interesting, we had two girls that we published a couple years ago that were sisters who both inherited an interstitial deletion from their mom, and they both had cardiomyopathy. So those are those two girls right here. So that really helped narrow the region. And so this is kind of how we map, and then we have to look and see how many genes are in this region. And so we can do the same thing with hearing loss, but then again, one of my frustrations is we found that it seemed like we had a region that was causing mild hearing loss, but then in the larger deletions here, this is the centromeres over here, so we're going towards the centromere. Larger deletions, we seem to have another critical region out here for the moderate to severe hearing loss. So it doesn't look like there's just one gene or one region that's causing the hearing loss. So the two sisters that I mentioned that we published recently, and both had non-compaction cardiomyopathy, one of them required medication, the other didn't, and they both improved over time. But that's a good question. When we're trying to do correlations between deletion sizes and the number of genes that are deleted and the clinical features, what we expect is a graph like this, that the smaller the deletion, the fewer the clinical features, and as the deletion gets larger, we expect to then have all of the clinical features. This is what we find with 1p36. The charts look a little different. We don't have good correlation between the number of the clinical features and the sizes of the deletions. And this is really the question I get asked the most, and I hear parents talking among themselves, well, how big's your kid's deletion? Well, how big's yours? And it's like, wow, he's done so great for, It's because we don't understand. We don't know. And so I want to finish up here because Bernice's research is really what I think hopefully going to help us understand this better. So done. Okay. So these results have led us to believe that we actually think that most of the genes that are most important in this syndrome are on the very end of the chromosome, where most of the kids are deleted. And that there's other factors that influence the expression or presentation of each feature. And that, again, is what Bernice her research focuses on. So does size matter? Well, for chromosomes, sometimes, not for 1p36. So for most chromosomal disorders, the smaller the deletion, the fewer the clinical features, and the better the outcomes or the development of the child for most chromosome disorders. And we have not observed this necessarily with deletion 1p36 or monosomy 1p36. We have some kids that have what I would consider quite large deletions that have very mild effects, and some kids that have quite small deletions that have very severe effects of that deletion. The smallest deletions that we've observed is about 200,000 base pairs in size. It's also called 200 kilobases in size. And the largest deletions that we see in the clinical lab are about 11 million base pairs in size, or 11 megabases in size. We've had kids in the study that have had a little bit larger than 11 million base pairs, but we think that much larger than that is probably lethal, and we just don't see it very often in the clinic or in the laboratory. So what it looks like is that it's not really the amount of DNA that's lost, but rather the location of the deletion or the location of the genes that are maybe causing the problems. We do use small deletions, though. They can help us at least narrow the region in which to search for genes. This was a recent paper that we published where we looked at very small deletions. These are the kids in the study. You can see that many of them have features that are consistent with 1p36. This is published, so I won't go through all the clinical features. But what we do then is, as I told you, we align the breakpoints and we look for the smallest region of overlap. But even with these kids who have very mild features, only 2 of the kids had seizures. They all had most of them had almost all of the facial features, and we're able to get this pretty small region on 1p36. And this is actually in 1p36.33. We see that there's a bunch of genes in there, and they're actually all good candidates for the phenotypes. This particular gene here is a good candidate for the seizure phenotype. The kids that had the larger deletions here had the seizures. These 2 patients didn't have seizures. And the ski gene, for those of you, we go way back. I think on my first talk to you guys, I talked about ski because mice, we worked with Clemencia colmenaris, and she knocked out the ski gene in this region. And the mice sort of looked like they had 1p36 with the facial features. They had large anterior fontanelles. So we think that maybe that's causing the facial features. But it's just been very difficult. And then recently, these are the 2 girls. So we published these 2 girls with the very small deletions who both had, they both had the non-compaction cardiomyopathy. Here are the 2 girls. So you can see that they do have some of the facial features of 1p36. They had fairly small interstitial deletions of 1.5 megabases in size. Again, mild features of the deletion, and both had the non-compaction cardiomyopathy. So neither required medication. I know that one had more severe than the other, but they both are improving. And they both have sensory neural hearing loss and delayed developmental milestones, so perhaps because they have such a small deletion, this will help us identify the genes. So even though these girls only have a very small deletion, they have 25 genes deleted in the region. And so this makes it very, very difficult for us. Their deletion actually doesn't start till they're almost 2 megabases in from the end of the chromosome, and extends till they're about 3 million base pairs from the end of the chromosome. And they may have mild features because we really think that it's probably the very end of the chromosome that's causing most of the features. And again, we looked at all the genes in the region. We actually sequenced all these genes. We didn't find any mutations in any of these genes. They're all missing in these girls. And they all, given the function of these genes, they all could be candidates for the cardiomyopathy. So again, very frustrating that we can't narrow it down more. And then we have 3 candidate genes for the hearing loss. So in summary, we keep calling monosomy 1p36 a newly recognized clinical syndrome. It's been 20 years, and I do think that some physicians are now recognizing it, and hopefully physicians have heard about it. It is unfortunate, though, that many primary care providers have never heard of it, and so you guys probably walk in and you hand them the medical literature on it so that they can get educated. But a better understanding of the syndrome will definitely lead to better treatment options for the various clinical features. So, I'm so happy that you guys have organized yourselves, and that there's studies going on, and it'll just improve these kids' lives. It's now easily identified using microarray technologies, and in fact, in the genetics world, very few chromosomes are done anymore. They're still done, which I don't understand, but most physicians are ordering microarrays when they suspect a chromosomal disorder, because a microarray, if it's there, it's going to find it. And I figure by now I've impressed upon you that this has been very difficult research. And the reason why the syndrome is so common is not well understood. We would like to understand why this deletion occurs, but as I showed you, it occurs through a variety of different mechanisms, and so that also complicates the story. And I will stop there, and I've answered some questions, and I think I'm way over. Okay.